Everyone, my name is Ronika Yousuf, and today I will talk about the absolute foundation of microbiology, culture methods. If you cannot grow a microorganism, you can't study it, test it, or identify it. This is where all our work begins. So, what is culture method? Simply put, it is how we trick microbes into growing and multiplying in a controlled lab environment. We provide them the perfect food, temperature, and habitat. The methods we cover today are essential for two things. First, isolation, getting a pure sample. And second, study, running experiments on a simple type of microbe. We start with the most common and the most crucial technique, the streak plate method. The primary purpose here is isolation. We use a loop to dilute a mixture of bacteria across the surface of an agar plate in a specific pattern. The goal is simple, to thin out the bacteria so much that the final streaks containing single separated cells. Each of those single cells will then grow into an individual visible pure colony. While the quadrant streaking is what you will use most often, there are actually several variations, and you need to know which one is to use when. The quadrant streaking method is our golden standard for high dilution. The tree streaking is a similar variation that also achieves great dilution. But if you need to quickly check if a sample is contaminated, a simple zigzag streaking pattern might be faster. We also have radiant streaking and semi-quantitative streaking used for more specialized counting purpose. They all achieve dilution, but through different patterns. Moving on to a technique with a completely different purpose, that is lawn culture. The goal here is not isolation, it's to create a uniform, dense, continuous layer of bacteria across the entire plate. We inoculate the plate and then incubate it usually at 37 degrees Celsius for 24 to 48 hours. This dense lawn is absolutely necessary for things like antibiotic sensitivity testing, where we need a constant layer of growth to observe the zone of inhibition around a disc. The pour plate method is one of our quantitative techniques. We use it to count the number of viable bacteria in a sample. Instead of streaking on the surface, we mix the bacterial sample directly into molten agar and then pour the entire mixture into the petri dish. When it solidifies, the bacteria are trapped within the agar, not just on the surface. This is a highly effective way to calculate colony forming units, or CFUs, in a liquid sample. Now we shift to methods focused on storage and maintenance. The slant culture, also called a slope culture, uses a test tube with an agar surface that has been angled to maximize the surface area. We inoculate it in a single straight line. Slant culture are primarily used for keeping stock cultures, our reference strains, viable and pure for short-term storage. The stab culture is similar, but it uses a deep tube of solid media. We literally stab an inoculating wire straight down the center. This is key for long-term storage as it protects the bacteria from drying out. Crucially, it's also used to study bacterial motility. If the bacteria spreads away from the stab line, you know they can swim. If you need large volume of bacteria, say for biochemical analysis or vaccine production, you use liquid culture or broth. We inoculate a sterile broth flask and often place it in a shaking incubator to ensure maximum aeration and nutrient availability. This method allows us to grow large quantities quickly and efficiently. The final section addresses the microorganisms that simply cannot survive in air, the anaerobes. These methods are all about excluding oxygen. This slide shows the specific media we use, like anaerobic blood agar and cooked meat medium. These media are specially formulated with components that reduce oxygen or provide key nutrients that facilitate anaerobic growth, which is crucial in clinical diagnosis. To actually create the oxygen-free environment, we rely on specialized equipments. I want you to remember these 4 key methods. The candle jar is a low-tech method where the flame consumes some oxygen. The gas pack uses a chemical reaction to rapidly consume the oxygen and generate CO2. The anaerobic jar is the sealed container that holds the plate in this oxygen-free atmosphere. And finally, the biological method uses other organisms to create the necessary conditions. Understanding this equipment is vital for successful anaerobic work. We finish with the spread plate method. Like the pour plate, it's used for enumeration, but it keeps the bacteria on the surface. We use a sterile L-shaped rod to evenly spread a small liquid sample across the agar surface. This results in individual non-overlapping colonies that are very easy to count, making it an excellent alternative to the pour plate for counting viable microorganisms. This was all about today's topic. Thank you for watching and thank you for being here.